The overall goal of this procedure is to demonstrate a novel laser-activated surgical adhesive called SurgiLux. This is accomplished by first preparing the SurgiLux adhesive solution. Next, the SurgiLux adhesive film is fabricated. Then, laser irradiation is used to bind the SurgiLux thin film adhesive to tissue. Finally, the adhesive bond is tested. Ultimately, results can be obtained that show SurgiLux thin film adhesive binds strongly to tissue after laser irradiation.
The main advantage of this technique over existing methods, like fibrin and cyanoacrylate surgical glues, is that the SurgiLux adhesive is a thin film that provides ease of operation, no curing time, and uniform bonding to tissue. Demonstrating this procedure will be Elizabeth Karsten, a PhD student in the Biopolymer research group.
To prepare the SurgiLux solution in a laminar flow hood, begin by making a 2% solution of acetic acid and deionized water. In a sterile Eppendorf tube, weigh the chromophore Indocyanine green, or ICG. Wrap the tube in foil to prevent any light penetration. Using a clean, disposable pipette, transfer approximately 1 milliliter of the dilute acetic acid solution to the tube and shake gently to dissolve the dye. Transfer the solubilized ICG into the beaker and add 2% Chitosan powder before adding a sterile magnetic stirrer. Cover the